pollinator. There are these environmental factors, indirect effects, and third-party individuals that can alter the fitness consequences for partners within a mutualism. And it's this variation in fitness outcomes that makes the stability and persistence of mutualism difficult to predict. One example of variability in fitness consequences within a pollination mutualism is the presence of secondary chemical compounds within floral nectar. So floral nectar often contains these secondary compounds. Often this is a byproduct of the herbivore defense mechanism, where chemicals make their way from plant tissues into the floral nectar and are then consumed by pollinators. And this nectar chemistry can have positive or negative effects on the plant or the pollinator. So for instance, from the plant perspective, there's evidence that floral nectar that contains some of these secondary compounds can improve pollinator constancy, which can have a positive effect on plant fitness. On the flip side, if your nectar is too toxic, pollinators can have trouble processing it, in which case you would, the plant would deter pollinators and there'd be a negative effect on plant fitness. From the pollinator perspective, having to process some toxic nectar can be physiologically costly, but there's also evidence that the presence of secondary chemicals in nectar can improve pollinator cognition and pollinator health. So the pollinators I'm focusing on in this talk are bumblebees, and so when we're talking about fitness of bumblebees, we're really talking about colony level fitness. So I've created a diagram here of the bumblebee colony life cycle, which is annual, and the reproductive unit of the colony is the queen. So when we're thinking about colony level fitness, we're thinking about queen survival and reproduction. However, we know that worker survival is really important. So we're talking about global change and mutualism, and it's probably no secret to anyone in this room that plant pollinator mutualisms are under threat of global change. And all these axes of global change are also going to alter the fitness consequences for plants and their pollinators. And today I'm specifically talking about the use of neonicotinoid pesticides. And so these are neurotoxins that also work their way into floral nectar when either seeds are treated with these pesticides or they're sprayed onto plants. And they work their way into plant tissues and then into the floral nectar. And this is true in plants in agricultural fields, but also non-target plants, so like wildflowers that would be growing adjacent to an agricultural field. The pesticide works its way through the soil, into the plant tissues, and into the nectar, where it's consumed by pollinators. And we do know that the fitness consequences of these neonics on bumblebees are generally negative. So we have these two lines of inquiry where we're understanding the effects of nectar secondary chemistry on bumblebee fitness and pesticides on bumblebee fitness, but we really know very little about their combined effects. And this is important because if a pesticide works its way into the nectar of a flower that already contains these chemical compounds, the pesticides are going to change that overall chemical structure. And we already have these variable outcomes when we're thinking about differences in sort of the concentration of nectar chemicals, and now we're throwing pesticides into the mix, and these outcomes may become even more variable. So this is what I and my collaborators are investigating, asking specifically here, how do dietary nectar chemicals influence how bees respond to an acute dose of a pesticide? And we can see this working in a number of different ways. So first, it may be that when bees consume these secondary compounds, it sort of primes their immune system or their physiology, their detoxification system in some way, so that when they consume then a dose of pesticide, they're more able to tolerate it. Conversely, processing of these compounds may be physiologically costly, in which case if they consume then a dose of a pesticide, it may increase the risk of toxicity for them. And I should add that these effects can be additive or synergistic, or they may cancel each other out. So we conducted an experiment using bombus impatiens, which is a commercially available bumblebee species used in agricultural settings and in research in the lab, where we measured survival and immune function in response to this combination of nectar secondary compounds and pesticides. So the way that worked is we set these up in these individual tubes and gave them sucrose feeders, so the feeder either contained just sugar and water, or sugar, water, and then the addition of some well-studied nectar secondary compounds. The compounds we chose were caffeine and thymol. So caffeine is found in citrus flowers, and thymol is found in wild thyme, cultivated thyme, oregano, things like that. And so they were fed on these diets for 3 days, at which point we gave half of the bees an acute dose of pesticide dissolved in a sugar solution, and half of the bees got just a sugar control. So we end up with this factorial design where bees either had just sucrose, sucrose followed by an acute dose of pesticide, sucrose with the nectar chemical, and then sucrose and the nectar chemical followed by an acute dose of the pesticide. We took a subset of those bees and we measured their immune function after 24 hours, and then with the rest of the bees, we put them back on that sucrose or sucrose plus chemical diet. We measured their daily consumption of those sucrose diets and then quantified survival under the different treatments. So first I'll tell you about the immune functioning response that we looked at. And so here on the y-axis we have phenol oxidase activity. And so phenol oxidase is an enzyme in the bee hemolymph, which is associated with immune function. And on the x-axis we have our different nectar treatments, so plain sucrose, sucrose with caffeine, and sucrose with thymol, and then broken down whether or not the bees had that acute dose of a pesticide. And some interesting things come out of this analysis. First, we found that bees that had caffeine in their diet had a little bit increased in their phenol oxidase activity than bees that had sucrose with thymol or just plain sucrose. And also when we compare within each of the sucrose treatments, whether or not bees had the pesticide, we see some conflicting patterns. So in the sucrose-only treatment, bees that had a dose of pesticide had a little bit of a boost if you compare those medians in their phenol oxidase activity, but in the two chemical treatments, bees that had an acute dose of pesticide had a little bit of reduction in their phenol oxidase activity. So now I'll tell you about survival, and I mentioned that we also measured consumption along with survival, so I wanted to put everything on the same plot. So now on the y-axis we have time to mortality in days, and on the x-axis we have daily nectar consumption, so milligrams of that sucrose solution we consume. We controlled for bee body size when we took that measurement. And then the pluses on the plot represent the sucrose treatment, the circles the caffeine, and the x's the thymol treatment. And so when we're only taking into consideration the nectar treatments, we don't see a ton of patterns emerging out of the data. There's a little bit of evidence that bees that consume thymol in the sucrose were a little bit less likely to survive than bees that consume caffeine or just plain sucrose. But patterns really start to emerge when we break it down by whether or not bees had that acute dose of a pesticide. So now I've color-coded the symbols where the bees in blue had an acute dose of pesticide, the bees in orange did not. And now we see this really strong pattern emerging between daily consumption and survival, but only for the bees that did not receive the dose of pesticide. So the more you eat per day, the longer you live, is kind of intuitive. But then the pattern totally goes away for bees that had that acute dose of the pesticide. And I think this is interesting because you see this same amount of variation in survival between pesticide-dosed bees and non-dosed bees. But you can have consumed a pesticide and survive on like no sucrose per day and live just as long as a bee that was eating 75 milligrams of sucrose per day. So we wanted to dig in a little bit into why we might be finding this pattern. And we know from the literature and also just watching these bees in the experiment that consuming the neonate pesticides really zaps their activity. And so Denise Marquez Sanchez, who is an author on this talk and an undergrad at Carleton College, did a behavioral assay where she looked at activity in response to these different chemical combinations. So the way that worked is she took the bees in their observation tubes and put them down over a dark line in the center of the tubes and just counted the number of times the bee crossed the line in a 5-minute period. And sure enough, the bees that were dosed with pesticide didn't move at all, so those are just zeros in the whole data set, versus bees that did not have a pesticide were more active. So, kind of trying to fit this all together and like stepping back and getting a little bit of perspective. We know worker survival is important for colony fitness, but it's really looking like there are these sort of interesting interactions between these dietary, physiological, and behavioral factors. So we know from the experiment that these dietary pesticides really reduce consumption and reduce activity. But one thing that would be interesting to think about is like the metabolic demands of bees that are moving around versus bees that aren't dosed with pesticide and are moving, and whether there's some sort of feedback here, and also how that plays into worker survival. We know that consumption has a positive effect on worker survival, but not when the bees have had that dose of a pesticide. So there's something going on in this pathway where investigating the role of the pesticide, I think, would be important. We know that dietary nectar compounds, in this case caffeine, can have a positive effect on the immune function. But one thing I'm really interested to dig into is sort of the cost of activating that immune system and whether there may be some trade-off where if you're sort of trying to handle too many toxins, you could put your immune system into overdrive and can ultimately be harmful. So just to summarize everything, caffeine does increase the immune response in this experiment, but that interaction between nectar chemistry and a pesticide is likely chemical dependent. An acute dose of pesticide negatively affects consumption, but not survival. And pesticide consumption inhibits activity. And there is this complex network of direct and indirect effects that we're excited to dig into more. So I'd like to thank especially Taylor, Ramneet, Jessica, and Ronnie, who are undergrads who helped out with this project, other folks at University of Nevada, Reno, who work for the bees. Thanks to Jeremy for organizing, and thank you. And I guess I have some time for questions.